actually have three, and hopefully they are, oh, I see them there. Hopefully they are getting ready to share slides. So these are the three speakers who are going to present their work from the Cell Biology Lab. This is DJ Park, Dushanth Narayan, and Daniel Yoshimura. And they're going to tell you about WD-40 containing protein expression during DNA damage. And I will let them take it away.
Let's get started. Hello, everyone. We are D&D, and we are representing the MCDB 3140 lab. We wanted to start things off by saying it is an honor to be able to give a live presentation at this year's Cure Symposium, and we also wanted to thank you all for tuning in tonight. Now, for the fall 2021 semester, we conducted an experiment in order to investigate our selected WD-40 containing protein's involvement in DNA damage repair based on its expression level in the protein. The first step of this thorough process was to understand what DNA damage response was. DNA damage response, or DDR for short, is an important network of cellular pathways in all organisms that is responsible for the production of proteins necessary for the survival of a cell. However, misregulation of these pathways can lead to serious issues such as loss of function in the cell, cell death, and even cancer. By continuing to study these pathways, we can gather enough evidence to potentially improve the overall quality of medicine and healthcare. The organism that we used in order to investigate our selected gene's involvement in DNA damage repair was Tetrahymena Thermophila. Tetrahymena Thermophila was the model organism used in this particular experiment due to the fact that it has been studied extensively and is known to be a safe and inexpensive organism that can be grown and bred in a laboratory setting. In addition, Tetrahymena Thermophila performs extensive DNA damage repair, which was the main focus of our study. This is a visual representation of our model organism during conjugation, which is the process of DNA being passed on from one cell to another using extensive DNA damage, as well as extensive DNA damage repair. Our selected gene's name was WD-40 containing protein, or WCOP40 for short, since it contained the WD-40 domain, which is basically a section of the protein that is known to be involved in DNA damage repair. These two diagrams showed the expression levels of messenger RNA and Rad51 and our selected gene during conjugation. Rad51 is a gene that is known to be involved in DNA damage repair, and since our selected gene contained a similar expression profile of mRNA as Rad51 during conjugation, it allowed us to ask the question, does gene expression change in our selected WCOP40 gene when induced with DNA damage? To answer this question, we conducted experiments involving using PCR tests, running gel electrophoresis, and designing primers, which Daniel will talk about next.
Right, so in order to study the gene expression of WCOP40, we first need to be able to amplify it to a workable level and then visualize it on a gel. To amplify it, we're going to use PCR. And the way this is done is by adding template, primers, and a polymerase to a thermocycler. In the thermocycler, the sample is increased to a temperature at which the template DNA will separate into two single-stranded pieces, and then the temperature is reduced so that a primer can anneal to these template strands. Finally, the polymerase will extend from the primers outwards along the template. After many cycles of this PCR, the section in between these two primers is exponentially amplified. A sample from the PCR can then be placed into a gel, and with an electrical current, the DNA is passed downwards through the gel. Shorter fragments have an easier time moving through this gel, and therefore will move farther down. This process allows us to visualize our DNA based on its length. So for our primers, primers are basically just single-stranded pieces of DNA that have complementary regions to a template. This complementary region allows them to anneal to specifically the template. We designed our primers specifically to anneal to this spot and this spot in our WCOP40 gene shown at the top. The area in between is the area we expect to be amplified by PCR. There should be 422 base pairs for gDNA and 204 base pairs for cDNA. The reason it's smaller for cDNA is because these blue introns will not be present. So after creating our primers, we needed to make sure that they actually worked. To do this, we performed a PCR and gel electrophoresis. So here are the results from our gel. On the left, we have our expected results. And at the top, you can see the gene that is being amplified. Below that, you can see the template. And on the left, we have our 100 base pair ladder. For WCOP40, again, we were expected bands at 204 and 422 base pairs. For ATPV0C6, which is just a housekeeping gene, and for our purposes, a positive control, we expect bands at 300 and 900 base pairs. Finally, for our negative control, we expect no bands at all because we are simply just using water as a template. In our experimental results, we see bands at approximately the same locations as our expected results, and we do not see any unexpected bands in our negative control or anywhere else in the gel. Since our experimental results are so similar to our expected results, we are confident to say that our primers anneal specifically to WCOP40. So since we are confident in our primers, we can then go on to measure gene expression, or the amount of mRNA of our gene in the cell. To do this, we take our Tetrahymena Thermophila and split it into two groups. One group is untreated and the other group is treated with hydroxyurea, which induces DNA damage in the cell. Afterwards, an RNA extraction can be done, and since RNA is difficult to work with, we perform a reverse transcription PCR, which will give us cDNA. cDNA can be used as a template along with the primers from the previous experiment in a PCR reaction. The PCR products can then be run on a gel, and we will expect a larger and brighter band when more cDNA was present, and a dimmer band when less cDNA was present. This allows us to see a difference in expression from our untreated and our plus hydroxyurea groups. So here are our results. At the top, we can see the gene that's being amplified. Below that, whether the DNA was untreated or treated with hydroxyurea, again, the DNA damaging agent, and whether reverse transcription was done. The plus indicates the presence of cDNA, and the minuses indicate an absence of cDNA. These minus lanes can be thought as negative controls. So for RAD51, which we're using as a positive control, since we know it's used in the DNA damage response, we expect an increased level of expression when DNA damage is induced. We can see that below with a brighter and thicker band compared to the untreated sample. For our loading controls, we used our housekeeping gene, and we should expect similar levels of expression even when DNA damage is induced, since of course it's a housekeeping gene. And as expected below, we see two similarly sized bands. Finally, for WCOP40, while we expected an increase of expression when DNA damage is induced, or results similar to RAD51, however, we do not get these expected results, and we have very similarly sized and bright bands, indicating that WCOP40 may not be involved in the DNA damage response. However, to more precisely see if there actually is a difference here, we then performed a qPCR, which Nishant will talk about next.
So a qPCR is a quantitative PCR, and it's a little different from a normal PCR. In a normal PCR, you take your sample after all cycles have been completed. However, in a quantitative PCR, you detect the level of cDNA after each cycle. So from this, we can get an expression profile and therefore expression ratios of our untreated sample and our DNA damaged sample. So here are the expression ratios of our three primers that we used. Our housekeeping gene, ATPV0C6, we expected little to no change in gene expression, and we can see that by the ratio, we see a very little change between the untreated and the hydroxyurea sample. Vice versa, we see a dramatic increase in expression of RAD51 during DNA damage, which again we see here. An interesting data point that we got was the expression ratio of our gene, which showed a considerable decrease of gene expression after DNA damage. So what do our PCRs tell us? Well, our RT-PCR showed no change in gene expression, while our quantitative PCR showed a noticeable decrease in gene expression. These conflicting results indicate that further experimentation needs to be done. However, from the data we have, we can either come to the conclusion that our gene is not involved in the DNA damage repair pathway, or is indirectly involved in the DNA damage repair pathway, which again requires further experimentation to confirm. Finally, we would like to thank our professor, Dr. Kristen Moore, our TA, Ryan Saye, and our LA, Carrie McMillan, for guiding us through this course. We'd also like to thank the MCDB department for hosting this lab and giving us the opportunity to perform our research. And I'd also like to thank the University of Colorado at Boulder for allowing us to present at this symposium. Thank you. Any questions?
Nice job. All right, so again, if anybody has questions, feel free to either use the hand raising function or to put them into the chat. I will try to keep an eye on chat. And all right, I see one question in the chat here. So from Tim, he said he sees slight bands in the first gel result. Is that contamination? So if you go back to that first gel, I think he's talking about in the water lanes.
No, those are primer dimers. So these primers sometimes anneal to each other and form a band at the very end. The fact that we only see primer dimers in our gDNA for our housekeeping gene and the water for our G-specific gene is actually pretty good result. We should have normal gels show primer dimers for basically all the lanes.
And he mentioned that he was looking specifically at the negative control, and maybe it looks like there's maybe a light band in the WCOP40 negative control. I think if I squint and tilt my head a little bit, I can see that.
Maybe right here.
Yeah, I think it's kind of challenging to see, but any thoughts on what that might be?
Well, since it is around the same base pair length as this band here, it could be just a very slight contamination from here. That's my best guess.
That's a good best guess. Let's see.
Do you think the ladder contaminated the brightness? Not really, because our ladder is isolated. We don't really add that to any other samples.
Great answer. And I have a question for you. So you're talking about kind of future directions that we need more study for this gene. Any thoughts on what any of those future directions might be? What you'd actually like to do to figure this out?
So first would be designing different primers. Next would be doing our experiment again because we got conflicting results. If we do get the result that our gene is changes expression during DNA damage, then we'd have to see if that's a direct involvement or an indirect involvement. Or if our gene isn't involved, we'd have to look for another one.
All right, we got one down here. Why is gDNA expressed lighter than the other bands in the primer design experiment?
We could just have a different amount of DNA in that specific well.